Topic of the lecture is pneumonia, and basically we're going to talk about what pneumonia is, how it is acquired, what's the difference between bacterial and viral pneumonias. So let's get on with it. So what is pneumonia? Pneumonia is an infection of the lung which can be of one lung or both lungs, and it is because of different kind of organisms. It can be because of bacteria, it can be because of viruses, it can be because of fungi, it can be because of anaerobic organisms. So all of these are reasons for bacteria, for pneumonia. So how are we to differentiate between them? The basic differentiation point among the pneumonias is there are typical pneumonias, there are atypical pneumonias. In typical pneumonias, basically the causative organism is bacteria. Because bacteria is going to get in there, bacteria is going to the alveolar level, it causes the inflammation of the alveolar level, and exudates and transudates start appearing, and with time fluid or pus accumulates in the alveoli, and for the patient it's very difficult to breathe. That patient will have sputum, yellow colored sputum, that patient will have cough, that patient will have fever. That are the typical symptoms of pneumonia. That's why they're called typical pneumonias. On the other hand, we have atypical pneumonias. Atypical pneumonias are caused by viruses. Atypical pneumonias are caused by pyogenic bacterium. Atypical pneumonias are caused by anaerobic organisms. So what's going to happen in atypical pneumonia? In atypical pneumonia, virus are going to get in there, or some bacterium, pyogenic bacterium is going to get in there. It's not going to go to the alveolar level. It's going to stop at the respiratory level, at the level of bronchi or at the level of bronchiole. It's going to insert itself in the membrane, and with time it's going to get invaded. With time it will destroy that cell. And when it will start destroying the interstitium, it will incorporate itself in that interstitium. It will incorporate itself in alveolar membranes. So that patient won't have that much of pus production, won't have that much of exudates and transudates, won't have that much of cough even. So patient won't be throwing off sputum. Patient will have just dry cough, mild fever. Patient, if you go for X-ray, chest X-ray, if that patient comes to you and complains of cough, and you suspect that he might be having pneumonia, and you go for chest X-ray will be clear. There will be no patchy consolidation. There will be no lobar consolidation. So you won't be suspecting pneumonia. That's why they're called atypical, because their behavior is totally different from that of typical pneumonias. So what organism cause the atypical pneumonia? Viruses, basically. So we're going to talk about which viruses are going to be the causative organism for causing this kind of pneumonia. So let's talk about them. So what are community-acquired viral pneumonias? The name gives it up. This name community, it means that you are going to get it from some other person who already has it. So it will happen in a bunch of people. It can happen in colleges, it can happen in schools, it can happen in military camps. So all of the places where a whole bunch of people are present and only one of them is infected, you are most likely to get it. So how are you going to get it? When that person who has this infection, who has this organism inside his lungs, is going to cough, or is going to talk to you, or is going to sneeze, so these viruses will come out in droplets. These are droplet infections. These organisms will come out in droplets, make their way to you, get inside of your system, get inside your nose, and travel all the way into your lungs and cause infection. So that's why they're called community-acquired viral pneumonias, that they are acquired from other persons. They are acquired when you get in touch with the other person. One more way to get community-acquired viral pneumonia is that if you get in contact with body secretions of that person. So you might be wondering which viruses are the basic viruses or the most probable viruses to get their way to you. So there are 4 basic viruses that we're going to talk about. First of all, it's influenza virus, then it's cytomegalovirus, then it's adenovirus, and then it's measles virus. So all of these viruses are going to cause pneumonia in the patient. So let's talk about them one by one. First of all, we have to know what a viral pneumonitis will look like on a histological slide. This is how it will look like. You can see it has involved all the interstitium. You can see there's no interstitium. You cannot appreciate the interstitium in this patient. You cannot appreciate the alveolar membranes in this patient, because this viral pneumonia is going to affect the interstitium. This viral pneumonia is going to affect the parenchyma. This viral pneumonia is going to affect the alveolar membranes. So let me compare it to the pathogenesis of bacterial pneumonia so that it might be very clear to you that they're totally different phenomena. What happens in bacterial pneumonia is that a bacterium gets inside of your system, goes directly to the alveolar level, uh, causes the inflammation, causes the infection. All of the inflammatory mediators, all of the cytokines, all of them helper cells, all of the neutrophils, eosinophils, they will come running, macrophages will come running, and they'll fight. They'll have that, uh, macrophages will try to eat them and they'll try to exert their negative effects on the system. And what happens in the results is that a lot of pus will form, a lot of transudates, exudates will form. Inflammation is happening again and again and again. So that will lead to the formation of fluid inside of your alveoli. Once fluid is built up, it's very difficult for a person to breathe because that place, that alveoli is not taking place in the gases exchange. And there's restriction in the lung. So in the bacterial pneumonia, it's going to affect the alveolar level. But in viral pneumonia, it's totally different. So let's know how it's different from bacterial pneumonia. First of all, an infectious agent will come through how? Will come through what? Inhalation. You will inhale those droplets. You will inhale those droplets coming off the person who is already infected with these agents. So this is a normal cell. This is a normal respiratory cell. This is a normal alveolar cell. What will happen is the entry of organism will happen. Infectious agents will come in and they're going to affect which parts, which structures of the cell. First of all, let's know that what structures are there to infect, what structures are there to destroy. They're type 2 pneumocytes, it's because it's alveolus, it's type 2 pneumocytes, it's capillary beds are there, type 1 pneumocytes are there. So it's going to get in there and it's going to affect all of these structures one by one or simultaneously. So what will happen? What happens is that infection of type 1 pneumocytes happen. They're going to destroy type 1 pneumocytes. Why they're going to affect pneumocytes? Let me tell you, pneumocytes type 1 and type 2 are very basic structure of the respiratory system. They're responsible for the production of surfactant. They're responsible for regeneration of the cell. They're the building blocks of the cell. So they're going to destroy them so that there is no regeneration. They're going to affect even the capillaries because they're going to affect the walls, because they're going to affect the membranes. And what's the closest to the capillary beds? Those membranes. Even if an alveoli is in contact with the blood vessel, what's between them? That membrane. If that membrane is affected, it will cause inflammation. And inflammation will happen, some organism will travel to the capillary beds and cause the inflammation of the capillaries. That's why the patient will have dyspnea. So after the infection of type 1 pneumocytes and specifically type 2 pneumocytes, what will happen? Interstitial pneumonitis, because all of the interstitium has been covered by all of this multicellularity. You can see all of these structures are healed, hyaline membrane is there, hyperplasia of type 2 pneumocytes happened, then congested and dilated capillaries because some viral organisms traveled through that and caused the dilation, caused the congestion of the capillaries. And interstitial edema and predominantly mononuclear exudate. What are mononuclear exudates? Mononuclear exudates, when a bacterial infection happens, who come running? Neutrophils, basophils, eosinophils. But if the viral infection is happening, who will come running? Helper T cells. Lymphocytes will come running. So they'll form this mononuclear exudate. So all of this is happening because of this infection. Type 1 pneumocytes got destroyed, type 2 pneumocytes have hyperplasia now, capillaries are now congested and dilated. So all of this is happening and this is leading to interstitial pneumonitis. So what will happen afterwards? Like in the bacteria, there was congestion at the first stage, and then red hepatization, then gray hepatization, and then the resolution. Just like that, in this stage, when we have got interstitial pneumonitis, either the patient will have interstitial fibrosis, which is very rare in this condition because they're mild, or it will get resolved, which is better chance of it because viral, all the viral diseases have some time. After that time, if you keep the patient hydrated, if you keep the patient on the best kind of medication, they'll just go away. So interstitial fibrosis is very rare in this condition. On the other hand, resolution will happen when all of these helper cells and all of these lymphocytes destroy the viral cause and get the type 2 pneumocytes back to its form, and they'll regenerate the whole cells. So in viral pneumonias, inhalation of the infectious agent will lead to what? First of all, it's going to be virus which is going to infect type 1 pneumocytes, which will lead to alveolar injury, and this will lead to interstitial pneumonitis. But we studied through details. Or it will be pyogenic bacterium. Pyogenic bacterium will do what? How it is different from viral infection, how it is different from viral pneumonitis. Because when pyogenic bacterium gets in, it causes acute inflammatory response to bacterium. There was no inflammation in this, but acute inflammatory response is going to happen due to the bacterium. Afterward, alveolar injury, not necessarily it's going to cause alveolar injury. And it will lead to intra-alveolar pneumonitis. It will affect those membranes between the alveoli. It's not affecting the alveoli. It's affecting the membranes between them. So that's how we differentiate between viral pneumonitis and viral pneumonitis caused by a virus and pyogenic bacterium. Now let's talk about those organisms or those viruses that we talked about that are highly probable to cause viral pneumonitis. First of all, we're going to talk about influenza infection. Influenza is that virus, influenza is that organism that scares countries and communities the most because it's that virus which can cause epidemics, which can cause pandemics. Its spread is very speedily. It spreads like fire inside the community. So what happens in the pathogenesis of the influenza infection? So let's talk about it. This is a normal cell. It has its receptor. This is cell membrane. Receptors are there to receive any kind of cell coming in. So when a person sneezes in your face, who already has that infection, when a person coughs in your face, who already has that infection, when you get in contact with the secretions of that person, who already has that, who already is suffering from influenza infection, what happens is that those droplets will come out of his nose, will come out of his mouth, which contain these viruses, which contains the influenza virus. So that influenza virus will travel to you. But as you can see, this influenza A virus is labeled. First, let me tell you what's the basic structure of influenza. The basic structure of influenza is that it has a nuclear material in its center. It's single-stranded RNA virus. It has its nuclear material in the center, which is surrounded by a protein core. That protein core is there to protect it when it shifts organism, when it goes from one organism to another organism. So protein core is there to protect that nuclear material that it doesn't get lost. After that, there's a whole covering of it. There's a whole capsule who surrounds the protein and that material, genetic material. That capsule is there so that it can get in contact with another organism. It can get in contact with another cell. And on that capsule, look at these structures. These are called protein spikes. These protein spikes are there. There are two types of these protein spikes, H type and N type. H type is called hemagglutinin and N type is called neuroaminidase. You have to remember these names. This is very important. Hemagglutinin and neuroaminidase. What's their function? Hemagglutinin, all of these green you can see, these are hemagglutinin. These are hemagglutinin spikes. Hemagglutinin spikes helps in invading the cell. And neuroaminidase, these red ones, helps to escape the cell. So we're going to talk about how they are going to make it happen so that we can understand why it's called influenza A virus, we have to know there are 17 hemagglutinin types. Up till now, due to the research, we know that there are 17 subtypes of hemagglutinin, and there are 9 subtypes of neuroimmunidase. So if it's hemagglutinin 1 and neuroimmunidase 1, it's called H1N1. And that's why it's called influenza A virus. If it's different, if hemagglutinin 5 is there and N1 is there, neuroimmunidase 1 is there, it's called bird flu virus. So influenza's subtypes are decided on the base of the protein spikes and core material present. So influenza A virus affects cows and humans. So we are going to discuss influenza A virus in detail because this is going to affect a human. This is what causes influenza infection inside a person and leads to pneumonia. So influenza A virus, meaning it has hemagglutinin 1 and neuroimmunidase 1 on the surface of it. So what happens is when a person sneezes, it gets inside of your system, it travels through your nose and back into pharynx and then into the respiratory membranes. It goes on and connects itself to a membrane. You can see M2, NA, HA, because it's neuroimmunidase 1, neuroimmunidase A, hemagglutinin 1, hemagglutinin A, and core material, obviously. So what happens is that adhesion will happen. How that adhesion will happen? Every cell membrane has receptor sites present through which it can communicate, through which it can cause the adhesion to something. These hemagglutinin cells are going to get there and adhere themselves with those receptors. They adhere themselves with those receptors, and then that's how they cause the endocytosis. Slowly they start moving in. They bind themselves like a lock, and then with time they cause the endocytosis. Cell will cause endocytosis. This structure, this influenza A virus will come in. So this is how hemagglutinin helped us, that it got stuck to that receptor and then endocytosis happened. So it helped us in invading the cell. That was the core function of the hemagglutinin protein spikes. So it has done its function. It has got us inside that cell. So what happens afterwards? Endocytosis has already happened. This cell, this influenza virus cell, will move towards the nucleus. From the cell membrane, it will move towards the nucleus. At the level of nucleus, it will open up. That capsule will open up. That cell membrane is going to open up. That protein coat is going to open up. And it will unleash that genetic material it has in its core. That genetic material goes to nucleus and it hijacks the nucleus. It hijacks the structure that is responsible for functioning of the cell. It hijacks the structure that is the energy source of the cell. After its hijacking, it gives off its core material, and many hundreds and thousands of copies are made in it. Replication happens inside the cell. Replication happens inside that nucleus. As there are thousands and thousands replicas are being made, some of those replicas will move out of the nucleus and go to ribosomes. What ribosomes does? Ribosomes is protein making structure, protein making organelle. So if a messenger RNA goes to ribosomes, it has its code on it that what proteins to produce. Ribosomes will start working for it. Ribosome will make all of them proteins which are required for the production of that virus again. So the proteins, which proteins do you think will ribosome be making? Hemagglutinin and neuroimmunidase. Because core material is already being replicated in here. So we need hemagglutinin and neuroimmunidase in very bigger amount, in very higher amount. So ribosomes will do that function for you. Ribosomes will multiplicate that. Ribosomes will produce those proteins which are required for this structure to happen. So all of this neuroimmunidase and hemagglutinin will be produced here, and afterwards it will move to Golgi apparatus. What is Golgi apparatus? Golgi apparatus is there to pick up these proteins and transform it into vesicles. They coat it, they transform it into vesicles, and those vesicles travel to the cell membrane, travel to the cell membrane of that structure. Here they start assembling. The assembly starts here. Why assembly is there? I mean, why doesn't it go to the nucleus? Because if it goes to the nucleus, it gets trapped. So all of the material that is required for another virus to be produced, for other virus to be generated, is right here under the cell membrane. So messenger RNA has done its work. So viral RNA will travel there, get there, and here all of them proteins are there, and we have the core structure, we have the protein to encode it. So here, after the assembly, another virus, another influenza virus will be generated. And that influenza virus will try to bud off. This influenza virus will try to leave the cell. Why it's leaving the cell? Because it has used all of its resources. It wants to infect another cell. It wants to infect much more cells. It wants to replicate. When it's budding off, what happens is that these receptors, which are already present on the cell membrane, these receptors which are present on the cell membrane, will catch that hemagglutinin. Why? Because hemagglutinin was attracted towards those receptors. That's how we got in the first place. So hemagglutinin will get attached to those receptors. It cannot bud off. So what happens is here the function of neuraminidase gets in. Neuraminidase breaks that bond and gets the cell out of here. It breaks that bond and causes the release of many viruses, many influenza viruses. So that's how they got inside the cell. They reproduced. They got the viral RNA inside the nucleus. It got replicated. Then messenger RNA was sent to the ribosomes. Ribosomes produced those proteins which were required. Ribosomes produce those hemagglutinin neuraminidases. Then those hemagglutinin neuraminidases were sent to the Golgi apparatus. That apparatus transformed them into vesicles and shifted them to the cell membrane. They waited for it. Viral RNA got there. Viral RNA got in the core. Protein core was produced, and then neuraminidase and hemagglutinin attached to the surface. And that's how they left the cell with the help of neuraminidases. That's the whole pathogenesis of influenza virus. That's how it gets inside of your cell, it replicates and it leaves the cell, destroys the cell in the process. So it will happen again and again and again and again until the symptoms come in, until there's a lot of damage happening. So what will happen if we see a slide of it? What will happen if a person is suffering from influenza infection? What will happen to the histological slide of it? This will happen. You can see a lot of hypercellularity and a lot of damage in here. We already talked about that capillaries will be congested and dilated. You can see that, and you can see there's no alveolar damage. There's no alveolar space being damaged. It's all about the intra-alveolar membranes. They're getting destroyed. They're getting the worst of it. This is how lung will be seen if a person has suffered from influenza virus and did not heal and did not recover. Next on, we're going to talk about cytomegalovirus pneumonia. How cytomegalovirus is going to cause a pneumonia. Cytomegaloviruses are basically going to affect those people who are immunocompromised. They can be immunocompromised because of many reasons. It can be a child. Cytomegalovirus can go to a child through placenta. So inside that, a guy, a boy, a child can get this disease transplacentally. What other source we have? A patient who's suffering from AIDS is already immunocompromised. That patient is going to get it. Elderly people who are above 65, they're going to get it. Or if a patient has any other chronic disease due to which immune system is down, that patient is going to get cytomegalovirus. What cytomegalovirus does, It's going to affect the cell the same way a viral cell is going to affect the cell. It's going to cause a destruction of them interstitium. It's going to cause a destruction of those alveolar membranes. So, what's there to differentiate it from other cells? What cytomegalovirus does is it increases the size of the cell. It causes hyperplasia of the cell. But with the cell, it also causes the increased size of the nucleus. And inside the nucleus, there's a structure and it looks like an owl eye. It looks like the eye of an owl. That's why it has been given this name, cytomegalovirus. This is how it will look on the radiological slide. You can see all of the interstitium being destroyed, and you can easily see all of the interstitium. There's no consolidation at alveolar level. You can see interstitium being destroyed. You can see alveolar membranes being destroyed. What happens if we see cytomegalovirus pneumonitis histological slide? I already told you it causes a hyperplasia of the cell, hyperplasia of the nucleus, and inside that it looks like an owl eye. You can look at that. Owl eye. That's why it's been given the name cytomegalovirus. Look at that. Infected alveolar cells are there. Nuclear inclusion is there. Look at that. Nuclear hyperplasia is happening. The size of the nucleus is too much and it looks like an owl eye. If we look at the lungs of the patient who has been suffering from cytomegalovirus, this is how it looks like. Look at that how black it got, how destroyed it is. And one point to notice is it didn't reach the terminal level. It didn't reach the alveolar spaces. It affected the interstitium. It affected the respiratory membranes. So next on, we have adenovirus pneumonitis. The main identification point that you're going to have in adenovirus pneumonia is that there are smudge cells with basophilic nuclear inclusions present inside of this. So this is the identification point if you look at a slide of adenovirus pneumonias. Pneumonias caused by adenoviruses. On a chest x-ray, you can see this is how the patient will look like. It mostly affects the lower lobe. So whole lobe is being affected and interstitium is being affected. And due to it, maybe there's some inclusion of water and stuff. There may be some effusion in that. So look at that. This is the adenovirus pneumonia. It's killing the interstitium. It's killing the intra-alveolar membranes. But you can also appreciate that whole lobe is not affected inside a virus. A virus doesn't affect the whole lobe. So what's happening right here? Actually, it's a secondary bacterial infection. I told you if a virus attacks your lungs, it compromises your immune system. Your immune system is compromised. So bacterial infection are going to come in. And which bacterial infection, which bacteria is most likely to cause secondary bacterial infection? It's going to be H. influenza. We talked about it. H. influenza will get in and cause a secondary bacterial infection. And it has affected the whole lobe. So this has gotten to the complicated stage of pneumonia in which a person is already suffering from viral pneumonia and secondary pneumonia is there because of a bacteria. So what happens, what's the pathophysiology of adenovirus pneumonia is that adenovirus is going to affect the following structure. First of all, it can affect the kidney, it can affect the eye, it can affect the intestine, it can affect the lungs. If the infection is active, upper respiratory tract is going to be affected for the first. It will start with upper respiratory tract infection, which will lead to mild disease, and it will be self-limiting because they have a probation period. They have a period in which they are very active and afterwards they just go off. But if the infection is of low respiratory tract, what will happen is that it's going to cause mild disease or self-limiting disease if the patient is immunocompetent, if the patient is healthy, if the patient doesn't have all those diseases which cause the immunodepression. What are those diseases? The AIDS, other SLEs, other diseases in which immune system is compromised. If the patient has a healthy body, a healthy immune system, whether the infection is upper respiratory tract infection, whether the infection is lower respiratory tract infection, it's going to get resolved in no time. So in which patients it's most likely to cause adenovirus pneumonia, children, military recruits. It causes acute respiratory disease and pneumonia in these patients. But if the patient is immunosuppressed, if the immune system is compromised already, if the patient is suffering from AIDS, if the patient is suffering from any other immunosuppressive disease, if the patient has already been through some chronic diseases, what will happen is pneumonitis. It's chronic form of pneumonia, and it will lead to fibrosis. If the infection is persistent or latent infection that happened again, what happens is that if the patient is immunosuppressed patient, reactivation will happen and pneumonitis will happen again. Because they had latent phase, they got pneumonitis again. And then E1A expression will happen in smokers, and that will lead to COPD. It's still debatable that if a patient is going to get persistent or latent infection, and if the patient is smokers and there's an expression, it's going to get COPD, chronic obstructive pulmonary disease. So that's what happens if adenovirus gets in. It's going to affect upper respiratory tract, it's going to affect lower respiratory tract. In upper respiratory tract, the disease is going to be self-limiting. It will resolve itself. It won't need any medication. It won't need any kind of help. But if the infection is of lower respiratory tract, it will get resolved if the patient is fine, if the patient is healthy, if the immune system is working. On the other hand, if the immune system is compromised, the patient will get pneumonitis. And they can go into dormant phase. These are viruses, they can go into dormant phase and they can later express themselves. The latent infection will happen, or the infection has been persistent. They have been persistently exposed to this virus. What will happen in that? Pneumonitis will happen. But if the patient is also a smoker, then it will lead to the expression of E1A, and that will lead to COPD. Next on, we have measles pneumonitis. What is measles? Measles is caused by measles virus. It mostly happens in children. Now that we have so many facilities, not many people get this disease. But when we didn't have any vaccination, when we didn't have any precautionary measures, almost all of the people used to get it in their childhood. What happens in measles? Patient is going to get maculopapular rash. All of the systems are affected. Inflammation happens. So how it is related to pneumonias? Measles virus is going to get in and cause measles pneumonitis. And the identification point is it is going to have multinucleated giant cell with eosinophilic inclusion. That's how you recognize that it's because of this pneumonia is because of the measles virus. It's not because of any other virus. It's because a person had measles, and it led to measles pneumonitis. It's actually a complication of measles. This is how you'll see it on a slide. This is how you'll see it on a slide, and this is how you see it on chest X-ray. Look at that. This inclusion. This inclusion is due to measles pneumonitis. So what will happen in measles pneumonitis? In early phase, you are going to get measles virus inside of your system, and it will lead to the cell and lung macrophages are there, and SLEM cells and DC cells are there. So they're going to react to this entry of the virus. But what's going to happen in the late phase? Maybe it gets resolved right around here. But if it's persistent and it didn't get resolved, what will happen? The tight junctions will happen in the lumen, and epitheliums will be swelled, and look at these junctions. All these junctions will swell, and this will lead to measles pneumonitis. Then we have pneumonias due to anaerobic organisms. Basic two anaerobic organisms that we're going to talk about is psittacosis and then anthrax pneumonia. Let's talk about psittacosis first. On X-ray, this is how it looks like. It's in patches. Look at that, look at that, look at that. It affects in patches. It has a patchy distribution. It is not diffused. In the pathology, this is how it will look like. Look at that, because this area was basically affected, so you can easily identify that this pneumonia happened because of psittacosis. And if it's seen on the slide, this is how you'll see it. All of these viruses present here. All of these anaerobic organisms present here and destroying the cells. So what happens in pathogenesis of psittacosis? Eucalyptus tree is a basic cause of it because it sends off spores. Birds come and sit on those trees, and birds excreta is there. So when eucalyptus tree is being contaminated with those feces of birds, what happens is when the tree is shaken, or due to air, or due to some reason, when there's wind, high wind or small wind or whatever, what happens is these spores will travel to a patient, or going to be a patient, going to travel to a person, and a person is going to inhale that. A person is going to inhale that because that person might be enjoying the weather. That person might be out. That person might be sitting underneath that tree. So inhalation leads to the entry of spores inside the lung. It will lead to alveolar level and contaminate them. So lodging will happen in the alveoli. It will not happen in respiratory cells in the bronchioles. It will not happen in bronchi. It will not happen in a nose. It will not happen in pharynx or whatever. It will go directly to the alveolar level. At the alveolar level, when the lodging has happened, what happens is that it disseminates into the blood. It disseminates into the blood through what? We talked about it, that some of them are going to travel from alveoli to the blood through those capillaries which are there to get oxygenated. That blood will be taken to heart, and that heart will pump the blood to the CNS, and that will lead to psittacosis. If you go for a culture of psittacosis, you will see a positive culture in this disease. Then we're going to talk about anthrax pneumonia, how anthrax pneumonia affects it. What happens is the toxin and anthrax is there, and it leads to the alveolar level, and afterwards anthrax spores germinate, and large spores are lodged in throat and windpipe, which leads to immune system activation. Immune system will come in, it will destroy some spores, but not all of them are destroyed. But other cells which are not destroyed are going to germinate in 1 to 60 days, producing more bacteria. So this will go on and on until the bacteria, the colony of the bacteria is so much that it starts to produce a toxin. And that toxin leads to hemorrhages and tissue decay. And smaller spores penetrate alveoli, and they produce the toxin there. They produce this effect there. They cause the hemorrhage there. They cause a tissue decay at the alveolar level. Now let's talk about hospital-acquired pneumonias. What are hospital-acquired pneumonias? They are also called nosocomial pneumonias. When a person is sick or ill and gets to the hospital, and he's there to get treated, but in spite of getting treated, he's going to get another disease. He's going to get pneumonia. And that pneumonia is very dangerous because it contains all those organisms which are not sensitive to most of the drugs we have. So you have to be very careful in the setting of the hospital to not get any disease from the hospital. So what are the causes of pneumonia? What are the basic sources of pneumonia inside a hospital? It can be because of the hands of a health care worker. That patient has been in contact with the pneumonia patient already, and that patient coughed at him or sneezed at him, and his hands contain that virus that contains those spores, contains that organism on the hand. And if that health worker is going to lay his hands on you or support you somehow, you're going to get those viruses inside of your system, and you are going to get pneumonia. eventually. Ventilator circuit can also be the cause of it, because if a patient who was predisposed to pneumonia already and was there on the ventilator, and somehow he just left or died or whatever, and you were put on the same ventilator circuit, you were put on the same setting which he was there, maybe you don't have pneumonia, but you're going to get it because those viruses are still inside that, because he was breathing through that. Biofilm of endotracheal tube. Endotracheal tube can be the basic reason of getting the pneumonia, because not all of them are sanitated enough. Maybe some of them have spores of them. Maybe some of them have them viruses on it. And if you go for endotracheal intubation or tube, it is going to cause the hospital-acquired pneumonia, or we also call it nosocomial pneumonia. Afterwards, it can be due to inhalation into lungs via endotracheal tube. You put an ETT and you start inhaling. That might be the basic reason, that might be the major reason to get it, because it's already contaminated. You are not supposed to do that. You're not supposed to breathe through that. So nosocomial pneumonias can be due to what? All of these were sources of pneumonia, sources of hospital-acquired pneumonia. But what are going to be the pathological cause of it? Use of broad-spectrum antibiotics. Then it can be because of chronic pulmonary disease. It can be because of reduction in gastric pH. When reduction in gastric pH will happen, what will happen? GERD will happen. GERD will come up. It will somehow get aspirated and lead to pneumonia, because many of the bacterial cultures will be there. Many of the bacterial colonies will be there. Then it can be because of intubation for respiratory support. We already talked about it. The other factors are also there. So all of these factors, which are going to the basic cause of pneumonia being caused in that patient, pneumonia being the pathological factor in that patient, that is going to happen. Oropharyngeal colonization will happen, or gastrointestinal colonization will happen. Both of them lead to the system through aspiration. If it's oropharyngeal colonization, patient will aspire and they will go through that. That virus will go through the laryngeal system and lead to alveolar level. And if a patient is having gastrointestinal colonization, that person may have GERD. That GERD will lead to some fluid inside of your throat, and you will aspire that fluid. This will lead to lung defenses failure, because lung defenses are now overcome. Now viruses are there, immune systems are gone because of this infection. So when lung defenses are overcome, what happens is nosocomial pneumonia. Patient who came to the hospital was already suffering from a disease. That person was already going through a lot. The immune system was already down to its knees. And then what we did, we introduced a new virus inside the system, a new bacteria in that system. And that led to nosocomial pneumonia. They're very dangerous, so you have to be very careful inside the hospital system. Neutropenia and critical ill diabetic patients and the patients of cancers are most likely to get it. Factors in net state of immune suppression, we already talked about it, that patients are already immunosuppressed. They don't know that they're going to get another one. So all of this leads to nosocomial pneumonia. Even the patient is healthy, he's going to get nosocomial pneumonia. If the patient is suffering from all these, he's going to get nosocomial pneumonia, as long as the patient has been exposed to this kind of organism, this kind of viruses or bacteria. Now let's talk about aspiration pneumonias. Which patients are going to get aspiration pneumonia? In a healthy individual, a patient who's suffering from GERD is going to get aspiration pneumonia. Fluid or that gas or that organism which was present in that came out, you aspirated that, it goes inside of your trachea, it goes inside of your lungs and causes the aspiration pneumonia. But other patients are more prone to this disease. Patients who are always lying, patients who are bedridden, patients who are on ETT, patients who are already on a respiratory support system. So this is a cross section of a trachea. In aspiration pneumonia, what happens is that you're going to aspire the fluid coming out of your stomach, and it is going to get in there and reach the alveolar level and cause an infection, cause a pneumonia, and the alveoli are going to get into this shape because all of this mucus and secretions are there. They cannot participate in gases exchange, not anymore. If they're not participating in gases exchange, it will be very difficult for a person to breathe. That person will get dyspnea will happen, shortness of breath will happen, and all of this is because of it, because all of these secretions which came out got inside of that and caused infection. All of this entered the alveolar level and caused the infection and caused the pneumonia, and now patient is not able to even breathe. And due to this accumulation of fluid, many other bacteria will start growing in. They have found the perfect environment for their growth. They'll colonize there. They'll cause the infection to reciprocate multiple times. So this is what a normal alveoli looks like. Look at that it's taking part in gas exchange all the way. All of these alveoli are taking part in the gas exchange. On the other hand, actually maybe 20 to 30% are taking part in the gas exchange. That's why the patient is going to get dyspnea and shortness of breath, and patient is going to complain that he has not been able to breathe because breathing is not happening, respiration is not happening, gases exchange is not happening whatsoever. So this will lead to pneumonia. Now let's talk about what will be seen on aspiration pneumonia's lung. If you see a lung who has been subjected to aspiration pneumonia, what will you see? You can see all the destruction there, all the fluid there. The lung has blackened, the surfaces are scarred. And look at that. This shows the level of the fluid. It shows the level of the infection that aspiration pneumonia patients are going to get. And all the interstitial areas are also being compromised. Now let's talk about what will happen to the morphological slide if a patient has been suffering from pneumonia for for far long. This is how the slide will show itself. Multicellularity inside the alveoli, inside the alveolar spaces, interstitium, because all of this has been happening for a very long time. And scarring will start. Ultimately, scarring will start. Ultimately, there will be focuses in which it won't be happening. Ultimately, there will be the formation of abscesses. There will be the formation of empyemas at the end. So all of it can happen if a patient has been subjected to chronic pneumonia. Maybe it has started with a viral pneumonia, but a secondary bacterial infection got in and affected the whole lobe, affected the whole liver, whole lung in the situation. So this is how you will define a chronic pneumonia slide if you look in the microscope. So what we discuss in this topic is that how a pneumonia is going to get in here, how a pneumonia is going to progress through an organism being outside of your system to getting inside of your system and affecting your whole body in different ways and getting to this state. So what we learned in this lecture is what is pneumonia. Obviously, it is an infection of the lung, which can affect one lung or both lungs or one lobe or both lobes or even the interstitium. We learned about the viruses that are going to be responsible for causing atypical pneumonia. We learned what typical pneumonia is. We learned what atypical pneumonia is. Typical pneumonias are those which have all of those symptoms, and atypical pneumonias are those which don't have these symptoms. Even the X-ray won't be showing off a pneumonia and no consolidation whatsoever. Then we learned about how the influenza virus is going to get in there and cause a pathogenesis, all of its systems, all of its pathogenesis, that how those neuroimmunidases and hemagglutinin are going to help them and invade the cell, how the nucleus is going to help them in replication, how the ribosomes are going to help them in production of the proteins, how the Golgi apparatus is going to help them in converting those proteins into the vesicles that are going to travel to the cell membrane and get accumulated there until it's time for a new virus to produce and bud off. Then we learned about adenovirus, how they're going to cause all of the pneumonias in patients who are immunosuppressed. Then we turn to cytomegalovirus, that how they're going to affect it, how the children are more susceptible to them, how their pathogenesis work. Then we talked about anaerobic pneumonias, how they're going to affect the lung system and lead to pneumonias. Then we learned about hospital-acquired pneumonias, that how they are the most fatal kind of pneumonias because they're not sensitive to many of the drugs. And how are you going to get them? And what's the pathogenesis of it? What are the basic reason of getting hospital-acquired pneumonia? And then we learned about aspiration pneumonia, that patients who are bedridden or patients who are suffering from GERD or disease like that are more subjected towards getting that kind of pneumonia. So at the end of this lecture, you should be able to differentiate between typical, atypical pneumonias, how the viral pneumonias work, what is their pathogenesis, how are they different from each other on histological basis, how they're different from each other on pathophysiological basis, and what is going to happen if you go through an X-ray of the patient. How can you differentiate that this patient is suffering from anaerobic organism and this patient is suffering from virus? So this is all. Thank you for watching sqadia.com.